I will now start leading or trying to moderate a discussion section among the four panelists. So the first thing that I'll do is I'll do my best to summarize the four talks that we had in this session. Try to see what there is in common, what overlaps there are, and how we can benefit the bioinformaticians that are watching us here, and how we can stimulate, I think, a community that is open to foster collaboration and to talk about science and develop this field of bioinformatics. So in the first talk, we heard about mathematical modeling of an interplay between flaviviruses and their natural defective genomes. The second part was about reconstructing dynamic potential for objects with observations of multiple features, data like single cell expression for differentiation of cells, aging of organisms, and how omics and biomedical data is used to understand metagenomics, and how that can be also applied for what Dr. Brodsky told us very well, very interestingly, the haplotypes and haplotypes of different viruses and phylogenomics. In the second talk of Dr. Raul Andino, we had mathematical modeling for public health. We have a very interesting talk where he led us through the use of different bioinformatics techniques and models to understand also many aspects of genomic variation in the viral genetic material. Particularly, we are all interested here in the current pandemic. So these mutations and the biological role that these mutations may have in, for instance, the spread and specificity between the ACE2 receptor and the spike protein in I am talking obviously about the SARS-CoV-2 virus. And then in the third talk, we had a very great description of single cell sequencing, and also the application of numerous bioinformatics tools in the platform that is available by Pine Biotech, and that was led by Dr. Julia Panov. Finally, we had Dr. DasSarma lead us through this very interesting tour on extremophiles and astrobiology. There's a huge field for discussion, in my opinion, about the biotechnological applications of both viruses, extremophiles, single cell sequencing, and also the algorithms for studying all of these subjects. So these are we have a common molecular biology background in all of these fields, but at the same time, we are talking about very different fields of application. Therefore, different kinds of backgrounds, different kinds of people are necessary to advance and provide the benefits in areas such as medicine, computer science, and communication. We have seen in the past year how communication, information, and education industries adapted quickly to the reality of distancing that was imposed by the SARS-CoV-2 pandemic. From this adaptation exercise, we see that there needs to be an effort due to we see that clearly because of the pandemic, and that has not ended yet in many countries. And we see that we all need to have some sort of adaptation to a technology-driven world or culture. So this dynamic imposes a need for education. And here we are we want to talk about bioinformatics education, a need for collaboration, and also a need for communication. So I wanted to again introduce our speakers. Dr. Raul Andino is a microbiologist and immunologist at the University of California, San Francisco. Dr. Leonid Brodsky is a professor of bioinformatics and biostatistics and the director of the Tauber Bioinformatics Research Center at the University of Haifa, Israel. Dr. Shiladitya DasSarma is a professor at the Institute of Marine and Environmental Technology at the University of Maryland School of Medicine. And Dr. Julia Panov is an associate director for the Tauber Bioinformatics Research Center in Israel. I hope that the panelists will provide us with their perspective on how to navigate this changing dynamic of data and algorithms in biological research. So I have some general questions here, but where is all of this data revolution taking us? And what have we learned from the pandemic? What have we learned that we need to adapt to this world that is more and more computational driven?
I can start, just break the ice here, but
Sure.
just say that you know, I think your question is directed to an answer, so I'm going to try to get that answer. And that is, you know, the pandemic demonstrated that it's important to collaborate and integrate. And you can start seeing now papers that has 30 authors on it. And I think that's interesting. You know, I don't know exactly what is the reason for this to happen, but in my personal experience, pandemic has been very disruptive, but disruptive in a positive sense. You know, it make us stop doing what we were doing and start focusing on a different problem and starting to interact with people that normally don't work in virology. And that has been a tremendous experience for us, you know, at least 2 or 3 new collaborators that I would never thought I would collaborate with. It was important. Now, how this omics, you know, gets there is what we've been discussing before. I think that the important thing, and I think in part the Pine Biotech platform help us to do that, is to have a friendly interface in which informaticians and experimentalists can converge. I think at the end of the day, you know, like it happened to me with Leonid, having a coffee in a nice place, it has, you know, a tremendous effect, transformative effect. But I think having a platform, an interface that is friendly for both experimentalism and informaticians is the next step in which this information can be exchanged using, you know, on the situation that we are right now, that maybe we're not able to travel as much as we used to.
Yeah. So if I could jump in for a minute. So to me, the difference between bioinformatics and biology is sort of semantic, because the real difference is between data and knowledge. And both bioinformatics and experimental work can lead to data, but we need to transform that into our understanding, whether it be COVID or the evolution of life. And so I think a meeting like this is really very, very nice to have people looking at everything from cancer, virology. Obviously, we're all concerned about COVID, and it's the overriding concern around the world. But as I mentioned, the Tom Brock was doing discovery science by going to Yellowstone and looking at, you know, thermal ponds for microbes and isolated thermus aquaticus from which we purified Taq polymerase, and then, Kerry Mullis and all developed PCR, and maybe PCR is not as important for sequencing, but it has been for very long, and it's probably still going to be critical for diagnostics. So that's where the interdisciplinary collaboration is so important, I think, and especially going forward because there's so much potential.
Can I add to that talk? So you mentioned Taq polymerase, how it has been so important for the development of sequencing back in the day when Sanger was more used than now. And you can see there how these extremophiles are very important for other areas. And I also wanted to highlight, for instance, the isolation of reverse transcriptases from viruses. So transforming, for instance, RNA into cDNA and being able to sequence that is on. I'm not entirely sure if it's correct to say that it's only possible because there are virologists that developed the understanding and the basic understanding of molecular biology on the virus and how they use their enzymes and all of these processes. So this is key nowadays for us to be able to sequence RNA, for us to be able to look at gene expression, just like Julia looks at RNA-seq, and I think many of you also do other sorts of sequencing as well.
Right. If I can jump in again, I'm sorry. So in addition, I think there are technologies that maybe are considered old now, but they were the mainstay of molecular biology, like restriction enzymes. Nathans and Smith and Arbor won the Nobel Prize for that in the 1970s, and it was the reason why we have molecular biology today. It would not have been possible. And of course, more recently, there are CRISPRs, and all of those came about through discoveries resulting from virologists and microbiologists interested in different types of microbes and analyzing their biology, and in the example that you gave, Gio, on the reverse transcriptases and David Baltimore and people like that. So yeah, so I think that it's really important to be able to do the basic science on what is, I consider, a natural resource. I mean, the microbes and the viruses that are out there, I know that some of them cause disease, but they're really a natural resource. And they have so many amazing molecules and features that we can then utilize for various useful applications.
If I can jump as well, not to kidnap the discussion, but I want to emphasize the thing because to me, it's obvious, and I think for every scientist, it's obvious that the reason why we are at this stage here is because serendipity has played an important role in advancing science. Nobody could anticipate that somebody study yogurt will discover CRISPR, right? And I think the wonderful ideas to look at the microorganisms world because there's a lot of information there. In my mind, the problem gets a little political here, but I think the problem is that the people that are leaders in science right now, they are trying to save as much money as possible and demonstrate that the money that is invested in science, every dollar results in an important result in a paper. And that is completely wrong. I think the science has to be wasted. The money has to be wasted in science. 95% of the money should go doing good science But most of these results should not result in anything except to say, my idea was wrong. Because that's what Popper, will say, the only thing you can do is prove yourself wrong. But the 5% that does not result in something negative will pay for everything 10 times. And I think is such an important concept. And we need to appeal to the Congress, appeal to the leaders and say, come on, you guys, invest in phages, invest in bacteria, do that kind of things, because you don't know what's going to be the brilliant mind coming up with a new algorithm. So,
talking about how we have generated so much data in the past years, I think we have developed a lot, the molecular biology and the biochemistry and all of these, what I call wet lab experiments. And now there's so much data out there. I think we are really at the point, and I think Elia was sharing earlier today how I came to my PhD program without a bioinformatics background, without a programming background. And I realized during my studies how important it is to develop these coding skills, these bioinformatics skills as a scientist, as a student. So I wanted to ask Dr. Brodsky, what do you think are important, how do you think a person that does not have necessarily a background in mathematics or statistics or programming, how can we develop that important background, which is the ability to look at data that has already been generated, that is sitting maybe on databases that can be accessed. For instance, now we have the GISAID database, which has, I think at this point, it should be like 3 million sequencing files available for COVID, SARS-CoV-2. And we have databases such as TCGA, the Gene Expression Omnibus, and there are initiatives in the United States, in Europe, and in Japan that store these data that have been produced along these years. And how can we gain more experience on analyzing this data?
Okay, from my perspective, there are two actually major directions to do that. One is new opportunities, which new technologies, computational technologies, are giving us. For instance, just recently, this Google artificial intelligence-based prediction of protein structures, generation of millions of protein structures. This is completely new opportunity for us, which can be used. Another thing is interface, human-friendly user interface. So with friendly user interface, theoretically, any person, any biologist or any student can operate with databases and with sophisticated algorithms in natural way. As Raul said, actually, this is a way for normal to collaborate, because previously biologist is just asking a bioinformatician to do computational work, and this is problematic because this bioinformatician doesn't know biology or not interested in the project and so on. If the biologist itself can work themselves, can work with a platform, with some computational platform, and do his analysis, computational analysis himself, it will be great. And actually, the same approach in education, I think, is also very promising. So this combination of new opportunities from computational technologies and user-friendly interface between computer and human are two key features for success, so to say, for success in education and success even in research.
Thank you. Julia, do you want to add something?
No.
Okay, I was just going to say, yes, it is very important to have the coding skills, I would say, to be able to analyze and process sequencing data. And I also wanted to ask, how is it? I don't know if any you have any experience in, maybe Julia, you have a little bit of experience in providing the tools for the user, providing the tool that the user is going to run their analysis in. How is, and I don't know if you have a coding background.
So maybe I'll answer. So my background is in neurobiology, and I have been doing bioinformatics work and analyzing data. Providing tools for the user requires, I think, again, collaboration of many parties, collaboration of software engineers on one side, mathematicians who actually come up with algorithms, and also biologists who will be the end user of this tool and who will try to use it and fail many times before this tool becomes actually usable and user-friendly. So from our experience, at least, it takes a while from the tool to become actually user-friendly. And of course, another important aspect, I think, is being able to communicate to the end user how exactly this tool is supposed to be used, giving him enough, not only just information or guides, but also examples of projects and questions that this tool or this a number of tools that we call pipeline can answer in different combinations, of course. From my experience, from our experience, it seems like it's again a question of collaboration, good communication between different people with different expertise that sometimes speak different languages. So also combining everybody together is another challenge that has to be met before the tool becomes really usable and becomes something that other people can actually use.
Perfect. I think I should
I would like to add in relation to education that actually this is self-education, it's becoming self-education. This is a project, this is a flexible platform, and a user which is trying to solve the problem with the platform. How to make the platform flexible and which will cover all possible aspects of the project. This is a question for development.
Perfect. Thank you so much. I generally try to make connections, possibly where people do not see. So I find it really interesting that Dr. DasSarma works with astrobiology and the development of life on Earth as a result from the interaction of Earth with the cosmos. And I find really interesting how previous to how molecules evolve to form a cell. And it's really interesting that, Julia, you work with single cell sequencing and how that is informative about the interaction of virus with human cells. So there is a question here about single cell sequencing. What I think we should give some background to maybe the comparisons of single cell and bulk RNA sequencing. Someone asked, how do pipelines for analysis of single cell RNA sequencing differs from bulk RNA sequencing?
Okay, so maybe I can take that. So yeah, the pipelines differ actually quite a lot. In bulk RNA sequencing, you're dealing usually with up to hundreds of samples at one time. I haven't seen a lot of projects that deal with thousands. And when you analyze the single cell or single nuclei data, of course, each cell is considered a sample. So you have thousands, if not tens of thousands of cells, which also come in bulk. So there is a big question of normalization of data and batch correction between different donors, for example, and different preparation methods of the actual samples. And also single cell or single nuclei sequencing can be divided into two parts. The first part is actually getting to the expression table, which is much more complicated because you have these barcodes, and depending on the technology actually used and machines for sequencing actually used, you either have only cell barcodes or you have barcodes barcodes that are for cells and barcodes for each mRNA. So this is one problem or challenge that needs to be addressed. And then, of course, different challenges remain, such as batch correction and normalization of data between samples, and when here I mean samples is just donors or tissues, and then a big bottleneck and challenge is annotation of different cell types or different clusters of cell. Actually understanding what these cells are is another challenge that does not exist in bulk RNA.
Okay, thank you so much. Another question that I saw in the chat was for Dr. DasSarma. How can we transfer a gene from, how can you transfer extremophile gene in a heterogeneous system? I guess the question is, how can we clone the gene in a mammalian cell, for instance?
Right. So there's actually, that's actually fairly straightforward because the most of the extremophilic genes don't have introns and they're pretty straightforward to express. I think the more difficult thing is to set up a genetic system in the extremophile, which is what we were able to do with the haloarchaeon. But in terms of expressing proteins in either in E. coli or in mammalian cells, they use the same genetic code. They are, maybe the proteins are more acidic, but the pangenome provides genes that are not present anywhere else. So whether you're doing metagenomics or whether you're doing single cell genomics or whether you're doing more traditional classical genome sequencing, the genes that you obtain are potentially valuable. And you addressed that point a bit earlier, and I was just thinking then that it used to be that it was necessary for students to generate data to analyze. Of course, I'm talking about the last century, so pardon my age, but there's just such an avalanche of data, and this is why we're talking about bioinformatics. What I'm afraid of is with 10 to the 12 cells and the continuing decrease in sequencing technology and also development of meta-sequencing and metatranscriptomics and so on, the rate of production of data is not going to decline. So it just becomes really important for how we're going to sort through all of this data to convert it into useful products and into knowledge. And so one of those is to express proteins. And there have been projects, even at NIH in the past, of trying to identify the functions of unknown genes by a pipeline of expression. But those projects have not, maybe Raul might know better about it. I have not seen those types of projects continuing. And it bothers me because there is just such a resource. It's a natural resource that's out there for the taking. But we need to allow students to come in and sort through it and pick through it and find what's useful and express that in a mammalian cell or yeast or wherever.
If I can jump a minute to comment on a couple things. One is something that you said, Shiladitya, about information could yield very beautiful pictures and complex pictures that impress people, but doesn't necessarily take you closer to knowledge. And the connection to knowledge is so important. This exercise, having a clear path, to understand and go through this data to really learn something new. And that's something that Julia Chasse that I really like is you getting data from different patients and you need to normalize that, right? And that is because this issue of noise is so relevant in bioinformatics. There is millions and millions of sequences, but those sequences do not represent the same thing because the sequences were obtained differently from different patients with different platform. The technical noise and the biological noise could really impede to see the signal. And I would challenge, all our young audience here that in biology, but in science in general, in order to see something, what you need to do is reduce the noise. If you want to see a star in a telescope, you need to polish your lens such that there is no imperfections on the images, so you can recognize a new star. I think it's the same thing here. You need to get noise to signal ratios in a way that allows you to see the things that you want to see. Sometimes you need to know what you're looking for. But I think this issue of noise is critical, and I think it's part of this exercise in bioinformatics. The other thing I wanted to just mention is that I'm so pleased to be part of this panel because in a way, it's very different group of people working in very different things, but there is common things, which is often we study viruses and we learn something is used for sequencing. Often we study bacteria and we learn something else that was very useful for something else. And I think that is a over theme here. And I would say, this idea of beneficial microorganisms is a very fertile ground for bioinformatics and to learn that. And the last thing I wanted to say, sometimes when I was very young, I was very afraid of approaching people that had been established and they, run centers and they handle millions of dollars or whatever. But I have a message for the young people here. Don't be afraid. We are eager to contact you, eager to work with you, eager to collaborate in a way, learn together. I think that would be great. If this happened in this symposium, that would be successful, right? A signal of success.
Let me add that from my perspective, bioinformatics is becoming three-dimensional. Structural things are extremely important. For instance, this blast story, which you presented us, orthologs through blast, it would be three-dimensional blast, local similarities, which may be also very important.
Yeah.
And to deal with huge amount of three-dimensional structures which are generated by artificial intelligence programs, this is a challenge again.